So now the room is yours.
Thank you, Yves. So it's a great pleasure to be here. I'm from Metz, so being in Luxembourg feels like coming home. Has a special feel. But I did my best to try to bring you something from California. There was some sun this morning. The temperature is not there yet, but at least I did bring some sun. Next trip I'll bring the temperature also. What I'm going to try to do in the next 45 minutes is try to show you that if we really think about some of the biological problems we're facing, and we try to build an approach that allows us to go beyond what comes with the black boxes biologists are using, we open the door to a new way of thinking about those problems, of new way of solving those problems, and hopefully new ways to look in the future at what can be done between the more quantitative science that we are familiar with, math, computer science, and the more, in quote, applied sciences, like biology. I always like, when I give a talk to computer science, I always like to start with, what does a biology do? A biologist does sort of all day long. Most biologists are experimentalists. And what happens is they have a framework in which they perform their research. They collect data. And today, they collect a huge amount of data. Once they have data, they need to process them. Processing means what? Processing means go through the data that I have and try to find some meaning to this data that connects maybe to the problem I'm facing or maybe opens new direction. So here is this learning process. Once we've gone through the learning process, comes a synthesizing step where we summarize what we've learned new and connect it with what we already knew. Finally, hopefully all these results allow us to predict what we can't see, go the step beyond, go towards what will tell us what's actually happening in the biological problem that we face and apply them to new problems. In parallel, how does a computer scientist see those questions? Well, at the level of collecting, a computer scientist thinks first about managing this data, allowing someone to retrieve the information that's in this data. At the level of learning comes the problem of mining. That's data mining, extracting knowledge from raw data. At the level of summarizing, there's a lot of question about visualization. We need support to visualize the information that comes as numbers. Finally, predicting is the whole field of machine learning, inference, prediction. Biology is changing. There are sort of new challenges that come into the picture that we need to deal with, that comes with the size of the data. Biologists always speak about big data. What does it mean for us, big data? It means if we deal with management of data, now we have some issues with the hardware that we have that may not be adapted to the size and velocity of the information that we are trying to process. At the level of machine learning, the more data they have, the more noise you have. So the more you need to think of process of dealing with that noise. The second thing is you increase the number of data, you increase the number of variables, your standard O of n cube algorithm may not work anymore. It's going to fail when n is around 1 million. There's no way you're going to apply it. In the summarization, I don't know if you've ever tried to visualize 1 million data points on a piece of paper, but depending on the color you use for the data point, your screen is going to look dark or white, or red or blue. Meaning, you can't apply Excel on 1 million data points and hope to see the information as you used to do when you had 100 data points. I always like, at the level of prediction, to get the biologists to understand that now that they're dealing with much more data than they used to, now that they're dealing with problems that are societal, they need to think that they can't just provide solutions independent of the world that surrounds them. So there's a big ethical issue that comes into the picture that we need to deal with. That's not what I'm going to deal with today. In some sense, if I summarize what it is that we computer scientists can bring to the biologist, if it's big data, we need to think about volume, velocity, and variety, the 3 V's that everybody mentions about big data. We need to think about complexity. Complexity comes in terms of, on a pure practical point of view, complexity of the algorithm that we're dealing with, but complexity also means how do we represent the system that we're interested in. We need to worry about reliability, integrity, validity, but we need to worry about quality of data. And the 2 topics I'll cover today covers 2 of these 4 aspects. I'll look at complexity, and I'll look at quality. For complexity, I look with dealing with very large system in what I call biogeometry. And you'll understand in a second what I mean by that. In terms of quality, I'll spend a fair amount of time discussing about the problem of clustering in the presence of noise. And I'll explain more what I mean by noise in these problems of clustering. Let me start with the first one, complexity, dealing with large systems. Let's look from the point of view of a structural biologist. 30 years ago, the first structure, experimental structure of DNA was published. Surprisingly, it was 30 years after the model by Watson and Crick had been proposed. That structure, 12 base pairs, approximately 250 atoms. That's what we could deal with in 1980s. Learning how the small piece of DNA looked like is interesting, but obviously we want to go further. We want to see how that piece of DNA looked like inside a nucleosome, which is one of the major substructures of chromosomes. That's bigger. Now that we start seeing what happens in nucleosome, maybe we can look at viruses. What's happening inside the capsid of HIV virus, of dengue virus, any viruses that relates where the support of information is DNA or RNA. Even for eukaryotes, how does the DNA organize itself within chromosomes? Now we are no more speaking about 250 atoms. We're speaking about millions of atoms, for which we don't even have the experimental data. We only see the envelope of where it is. So that complexity that comes in size and in the fact that when we look at pictures of where the chromosomes are, instead of seeing the atom, we only see blobs that includes a cluster of atoms. When we try to deal with that quantitatively, we cannot try to fit atom inside. We need to think the way the data brings us the information. So there's a whole field of trying to represent those larger structures, not using an atomistic detailed picture of what's happening, but using a coarse-grained model where we have blobs that captures the local geometry that we have. And the way these blobs organize in space is the system that we consider. What's the most standard blob that we would like to use in geometry, because it seems that it's the blob that we know the most? It's the sphere. And if you think about sphere, what's fascinating is atoms are usually represented by sphere already. And at the larger scale, those different chromosome units are just collection of spheres. Spheres are extremely common in material sciences because that's the way you model granularity in material sciences. Sphere is, in some sense, is the bit of computational geometry. It's the bit of representation of shapes in biology. Spheres are nice because I can give you the surface area of a sphere, the volume of a sphere, without having to think. I was taught that in middle school. I can, in high school, take two spheres and give you the surface of interactions of two spheres. If you give me three spheres, the first paper was 1982. If I give you four spheres, the first paper was 1995. If I give you n spheres, how do I measure n spheres? Well, it's only sort of 10 years ago that we started having models about what's happening for spheres. What's the real problem? And how do we approach that problem? Think about it this way. For visualization, it's easier for me to show you a collection of disks. And I'm asking you, can you find the surface area that's covered by this union of disks, the red surface area? Well, the problem is you may have 20 disks overlapping. What's the surface area of intersection of 20 disks? I don't know how to do that. However, here's what I will do. I will use geometry to solve that problem, but slightly in a different way of looking at geometry. The first thing I do is, let's forget about the disks themselves. Let me only look at the position of the centers of the disk. And the first thing I build is, I have a small movie for you. I build, as soon as I get my cursor here, where is it? On? Here is the movie. The concept of Delaunay triangulation is you want to partition the space with triangles. Triangles where the vertices of the triangles are the atoms or the centers of the disk that I'm interested in, so that each triangle is, in quote, empty. Meaning, if I draw the circle of that triangle, it does not contain any other points that corresponds to that system of points that I'm interested in. The way you do that is there's an algorithm for doing that. You randomize the point, you locate the point inside a triangle, you subdivide that triangle, and you correct and flip the triangles that do not match this emptiness criteria. One thing that's important for computer scientists is the line named 1, randomize the point. Randomizing the point guarantee that in the worst case, the algorithm is O of n squared, and in most practical case, the algorithm is O of n log n. You have the guarantee only comes if you randomize the point. Once you have the Delaunay, the dual of the Delaunay, the dual structure of the Delaunay is the Voronoi decomposition of the space. What does it mean, Voronoi decomposition? If I look, for example, at that point, the Voronoi region around that point is the collection of all points that are closer to that point than to any other point in my system. Notice that the Voronoi goes to infinity. It covers the whole space. My last step in my approach to deal with that is to limit my Voronoi to only the collection of balls that I'm interested in. Now, what is interesting with that picture is when I look at that atom, for example, or that atom over there, The first thing I see is the total surface area that I'm interested in that's covered by red is now the sum of the contribution of each Voronoi region limited to the sphere that I'm interested in. So I was able to build a geometric structure on the top of my union of disks that exactly give me the measure of the union of disks that I'm interested in. The second thing that I'm really interested in is when I look at any of those spheres and I try to find what is its surface area, its contribution to, say, the surface area of the volume when I'm in space, that contribution is the total surface area of the disk times the fraction that strictly belong to that disk. How do I compute that fraction? Well, what I notice is, let me go to a very simple case where I have 4 disks. I use inclusion-exclusion. This is basic statistics, but my inclusion-exclusion is limited to term up to order 4 because of the Delaunay structure that I have in the background. So I guarantee that I can do an exact calculation here, that it is finite, and that is easily implemented. And the inclusion-exclusion formula just tells me that the total surface, the contribution of AI to the total surface of the union of disk is the total surface of the disk minus all the pairwise interaction. But I need to bring back the triplet because I removed them twice. And I need to subtract back the 4th intersection because I've introduced them back twice when I introduced the triplet. Because I only have to go up to level 4, the formula for computing surface area of this red region are very easily computed and gives very nice results. That's a Delaunay approach to measuring a union of disk or balls of any number n of balls, whatever the number of intersections that I have, and I'm guaranteed to only use an inclusion-exclusion formula up to level 4. Now I go back to big data. Oh, before that, just one thing. I won't go into too much detail about that. When I said here that I limited my Voronoi to only the union of disk, there are regions that are not included in those, that are not covered by the union of disk. What do these regions correspond to? These regions are very interesting because these regions are the cavities that exist within my union of balls. So what happens is not only can I measure the surface of the gray area, but I can measure also and detect those cavities that exist in my union of balls. That came as a side product at no cost in the algorithm. Why is that nice? When we started working on that, the first thing the biologist was saying, but can you show us the pockets inside the structure? So we took HIV protease, and we said, that's the structure of HIV protease on the left. This is where we see the major pocket. And then they have a structure of HIV protease in the presence of a drug that inhibits HIV protease. Well, that drug fits exactly in that pocket. So that means now they have a tool that tells them where they should look for regions where ligand could bind. I'll show you another example. This is ribosomal RNA. This, when the first structure of ribosome came out in 2001, it excited the drug companies to a very large, it created a new field, in some sense, for drug design. The reason being, ribosomes are a common element that exists in all cells. including bacteria. So if you're able to distinguish the specificity of ribosomes in bacteria compared to ribosomes in eukaryotes, you're able to target specifically something that you know exists in your bacteria, but you'll protect at the same time your eukaryotic ribosome. Within a year after the structure of ribosome came out, 45 structures of ribosomes with ligand came out. It's still an open field because they haven't yet found a ligand that is actually sensitive, specific, and antibacterial enough to be used for drug, but they're still looking actively in that field. Just to show you something, here is one case of a structure of ribosome RNA with the presence of the ligand. What I did was the same thing. I said, let me show where the pocket is in that big molecule. That's the green region that is there. But remember, I can increase the size of my sphere. I can play. Now I have a mathematical model. If I increase the size of my sphere, what's going to happen is what's going to remain at the deepest pocket in my system. And when you reduce the size, the deepest pocket is where the drug went and bind. The mathematics of doing what's on the left costs you 20 seconds on a computer. And you can try, you can play, you can do whatever you want. 20 seconds on a computer, except any computer scientist would go back to the algorithm and say, it's a beautiful algorithm. I have the math behind it that tells me what its complexity is. Let me apply it. So I apply it on a very small system. And then I increase the size of the system. Now I go towards these viruses that I was talking about at the beginning. And now I look at the computing time I get. And that computing time is definitely not n. Not n log n. Not even n squared. So the theory tells me it should be n squared. In practice, I'm far from being n squared. So what's happening here? People that do software engineering, first question they should ask is, do you have thrashing happening here? So what is happening here is the geometric structure that stores your Delaunay becomes spread over the memory. And what happens is every time you insert a new point, you're going to call back information coming from multiple parts of the memory. The cache becomes completely useless. And then you pay a very high price in terms of thrashing. The problem is spreading of the information. How do you solve it? You try to make the information as local as possible. So the easiest thing, this was in some sense in terms of algorithmic development, this is the easiest trick you can do, is you remove the line randomized point and you just replace it by a range point locally. In theory, you lose the guarantee that the algorithm is n squared. But in practice, the algorithm becomes n log n again. You remove the randomization, you remove the thrashing. It's nothing to do with theory. Sometimes, as a theorist, I have to adapt to the reality of the system that I'm interested in. And this is one really nice example of that. Just to show you, this is a system with 1 million atoms. You can detect the region that's covered by the atom. You can detect the different pockets that you have. So there's pocket in the membrane that gives permeation to inside of the viral capsid. And you have the very large green pocket inside. You can measure it, you can quantify it, you can play with it. Let me move now to the second aspect of my talk, which is, it's great to have data, but sometimes these data are noisy. Where probably we see that happening a lot in biology is when we have noise, we have a hard time clustering. Why do biologists clusters? Biologists always cluster their data. Why? Because it's easier to think about a representative than to embrace the information of all individuals. You think about population. When you try to think about population, you hope that all members of the same community will share the same properties. And you hope that whatever method you use to pull together these objects, it will detect those communities. But what if there is noise? What's a good distance measure to use? What's the measure of similarity between the objects that you can use? How to handle non-convexity? If you look at these two moons, they're not convex. So what's going to happen if you just take a system out of the box and try it? How to handle very large data set? I'm going to look more specifically at these 2 points. What's the good distance measure and how to deal with non-convexity? Just skip this. Just to show you, I gave that data set to a biologist and I say cluster them. So he went to the system he had, he applied hierarchical clustering the way he has always been used, and he cut the tree, gets a different level, and that's what he got. Even at the level of 2 clusters, well, hierarchical clustering finds convexity. It's looking for convexity. So it defines a plane here, and whatever is above the plane is in one cluster, what's below that plane is in second cluster. It did not capture the geometry of my system. And that's 90% of the biologists, that's what they're going to do, and they stop there. So what else can we do? How can we improve that? The whole idea is how can we capture the geometry of that set of points, knowing that that geometry is currently represented by a distance measure that is purely empirical, that for which I don't even have a proof that it's actually a metric. I just have an empirical distance measure. So let me try to capture something here. And then we go back to physics. And physics tells us, well, why don't you build a potential on your system? And that potential is going to say that if you are on data point I, the probability that you're going to build a path that takes you to a data point j is going to be simply proportional to exponential minus the distance you've established between the 2 points divided by temperature T, a factor that you have. Now that you have that potential field, you can build a Markov chain on it. You can use Monte Carlo to explore that potential surface. And what are you going to observe? Well, you're going to observe that at the beginning, if you start from one of these points on the left, it's going to mostly explore the data points that are in the neighborhood of that point. The cost of jumping to another cluster is too high. So you do that process, and you monitor this energy, which allows for jump locally. And then, in your process, you bias a little bit your Monte Carlo. If you've explored the data point n times, you remove it. You know it was in that local neighborhood. At some point, there won't be any red points to explore. So suddenly, you're going to have a big jump to a different region. And now you're going to explore that blue regions. And then you keep doing that. You're going to explore your black region. Once you finish that, based on the curve that you have for the energy, you're able to detect phase transition that occur at this specific situation. And then you build a connectivity matrix between the data point, where if 2 points were explored in that red region, they'll have a 1. But if the point went in between one of these red and the blue, for example, it'll have a 0. So you're going to have a connectivity matrix between the point. However, we know that that connectivity matrix is going to be highly dependent on the temperature T that you choose. If you choose a very high temperature, everyone is going to be connected with everyone. If you choose a very low temperature, every data point is going to become an independent cluster. The beauty of that method is you can build now different connectivity matrix at different time t. Once you've built those different connectivity matrix, for each pair of points, you just store the information at increasing temperature. Did they belong to the same cluster or not? At some point, because you go to very high temperature, at some point they are going to belong to some cluster. They may switch, belong to the same cluster, do not belong to the same cluster, re-belong to the same cluster. Doesn't matter. That's noise that you have in your system. You stabilize the system by defining a quantity u between two data points I and j as being the first temperature starting where all the connectivity matrices after that temperature have a 1 for that pair of points. The beauty is mathematically, you can show that the uij set an ultrametric on your space. So you get more than just a metric, you have an ultrametric now on your set of points. Why is it so nice to have an ultrametric? Let me go back to the data that I had at the beginning. I start with exactly the same Euclidean distance between the points, but I correct this Euclidean distance to build my ultrametric on my space of data points, and I recluster using hierarchical clustering. I didn't have to change the method, I just changed the distance measure. That distance measure is built from the empirical distance measure, and what I observe now is the clusters I find follow the geometry of the system that I'm interested in. Let me show you another application of that. If there's one thing that biologists are really interested in currently, is network biology. A representation of the interaction that occurs between the species they are looking at. This could be all the yellow data points that I have on the top, could be a collection of proteins inside the cell, and every time I know two proteins interact, I'm building a line between the two. Very common to represent interactions in biology using those network representation. Now, if those networks were only there to capture visually what happened between species, I don't need them. There are many other ways to represent that. My hope is I can use those networks to infer information about the system. And there are many ways to do that. You can build random trees that follows the network so that you can capture some statistics on your set of points. Now you're going to understand what will be the cost of removing an edge or adding an edge. You'll be able to understand what's the dynamic of your network. But to do that, you need to know what the network means. You have to capture its geometry. How do you do that? Because if you sample randomly on all the vertices that you have, or on sub-communities of the network, you're going to get very different statistics on the data. Ultimately, you want a statistics that capture the actual hierarchical organization of the set of points. How can we do that? Well, the method that we propose to do that is to remember that with this method I described for clustering, which we call data cloud geometry, we're able to capture communities. So what we do is we start with all the data points, we cluster them, give them colors, and then we organize this information into a Parisi matrix. That's a complicated term to say a connectivity matrix, but a connectivity matrix that has been reorganized to capture most of the information along the diagonal and not too much information outside of the diagonal. The thing is, just building that DCG-tree does not guarantee that I have a matrix that is as diagonal as possible. I need one more step in the process. That one more step, you can understand it like this. This on the left is the original information where you put a blue dot where two vertex are connected in a network, and a yellow dot when they're not connected. That's the original matrix. The matrix in the middle is the one that came out of the clustering. How is it related to the original matrix? Mathematically, you just apply on the adjacency matrix, you just apply a permutation. Now, what you really need is find the permutation that will lead to a system where all the information has been really gathered hierarchically along the diagonal. How can you go from that matrix to this one? Well, statistical mechanics. You want a system that represents the ground state of your system. That's the one where most of the information is along the diagonal. So you build a 2D Ising model on your system. So you build an Ising energy, a ferromagnetic Ising energy that depends on interactions between neighbors, with some terms here that corresponds to the original matrix after some permutation U. And you find the permutation U that leads to the minimum of that energy. You do that on the system that I have showed you. Let me show here a very toy representation of the system. I can't remember how many vertices there are. There are 420, so there are 84 vertices. You can think of the network between them as either a network with a single community or a network with 4 communities. Ultimately, you would like it to be with 5 communities, but just for illustration, I showed you the system with 1 and 4 community. If you randomly sample your network to satisfy a single community, you're going to find the random network. I should have gone into the detail a little bit. Random network with the same number of connections per vertex than the original network. But if you do not take into account the geometry of the system, this is what you're going to observe on the right. However, if you randomize according to the organization of the adjacency matrix that comes from the Ising model I just described, you will find this random network on the left. Now, I want to emphasize that in both networks, each vertex have, on average, I mean, obviously you repeat it, the same number of neighbors than the original network. The only thing is, once you're going to do your statistics on this one, your statistics are going to inherently capture the local organization of your data point into these 4, I use 4 here, into these 4 different regions. That means here, I'll go back 1 second to that. Here. That means what I try to do very pragmatically, I didn't go into the math detail of it, but what matters is really the concept behind this mathematics. What I wanted to show you is if you have any network and you would like to sample that network in a way that you capture the local geometry of your system, the way you will do that is you detect the communities in your network using a clustering method, and obviously the better the clustering method you're going to use, the more reliable the communities will be. Once you have detected these communities, you build an optimized adjacency matrix, a connectivity matrix, that has captured, shown on the right, that has captured all the information as much as possible along the diagonal. The interesting thing is once you have that adjacency, optimized adjacency matrix, now you can detect what is the level at which you want to capture your geometry. If your level is level 4 here, you're going to sample first along each of these blocks to build the set of edges that capture that local geometry. You may have off-diagonal terms that still comes in. So if you had 2 communities, you would look at that whole submatrix here, where you would do your sampling here, and you would look at each of the non-diagonal term, and you would add a few random edges that corresponds to those. But the number of non-diagonal terms, non-block diagonal terms that you're going to include will be minimal and will correspond to the local geometry of the network that you were interested in. So here is a mathematical method with an implementation that allows a biologist to think about this network with its hierarchical organization. That means go beyond just the black book bootstrapping method that comes with hierarchical clustering. So because I'm reaching the end of my talk, I just want to briefly summarize the key elements I wanted to get through within that seminar. An introduction to big data that represents size, velocity, but for us computer scientists, I believe the most important element that we need to deal with when we think about big data is the complexity. The cost when trying to deal with big data is that the traditional algorithms that we have may fail. We need to rethink the way we define local organization. We need to rethink the way we measure in those systems. Still, in the introduction, I emphasize that large amount of data means more noise to deal with. The key message that I wanted to emphasize with the clustering method is that you can rescue traditional clustering method by correcting the distance measure that you use to compare points. And the key is the way you correct that distance measure is by building a structure on your data point that naturally capture the local geometry of the points that you're interested in. So you let the system tell you what the communities are, and then you have a mathematical result in the background that tells you that if you do that carefully, you get an ultrametric, and then your clustering algorithm are going to be much more performant on the system of points. That was all part of a system that we called data cloud geometry. Is the ability to capture the geometry of data cloud, whatever the dimension of the system is. If any data point is represented by 1 million features, that's all captured in that empirical distance measure that you give. So we work in the space of distances, not in the space of features. That allows us to deal with a complexity that other methods would have a hard time with. PCA would not be able to deal with that very large size system. Let me end by telling you with whom I've been doing part of this work. There's lots of people with whom I collaborate. My main collaborators are Michael Levitt, Stanford University, who really introduced the concept of computational tools for structural biology. That was probably the big relief for the biophysics community was 2 years ago when finally someone computational got a Nobel Prize in chemistry. So that's, I worked with him for 10 years. When I started my PhD, I worked with a theoretical physicist called Marc Delarue, who is at Institut Pasteur in Paris. We have 20 publications together. Distance has never stopped us from working together. Herbert Edelsbrunner is the mathematician who introduced me to the concept of spheres and who showed me, we spent many hours, evening hours discussing. He showed me how to go from pure math to computational geometry and all the problems that comes with that. When you build a Delaunay, the problem is noise in the data point. Think about it. The most common predicates that you use when you build a Delaunay is you have a plane, you have a point, and you need to know if that point is above the plane, below the plane, or in the plane. How do you do that? In mathematics, you compute a 4x4 determinant. If the determinant is positive, you're above the plane. If the determinant is negative, you're below the plane. If the determinant is 0, you're on the plane. Anyone who has done calculation with floating point numbers say that you never get 0. You get sometimes 2 × 10⁻16. Do you trust the sign in front? So we build a whole robust system for dealing with that type of noise where you transfer the floating point into integers, and you do arbitrary precision arithmetics, and you do Taylor expansion when you're around 0 to find what is the actual sign of the determinant. At the bottom, I put the graduate students that contributed the most to that work. The guy on the left is the one that actually worked on the geometry. Fascinating is at the time he was doing his PhD, he was a professional biker doing the Tour of California and wanted to do the Tour de France, but an amazing mathematician. The 4 other students are students that we shared with Fuxing Xie, who is a statistics professor at UC Davis, where we have developed all these techniques that I mentioned about clustering. I'm not allowed, rightfully, to end the talk without mentioning who gives me some money to do that. So the NIH has given me money, the NSF has given me some of money. I'm a Sloan Fellow, so they gave me money for doing that. I'm a visiting professor of the National University of Singapore, so they gave me some money to do that type of work. But most importantly, you cannot finish a talk before thanking the people that listened to you for 45 minutes and didn't fall asleep at 2 in the afternoon after a very heavy and really nice meal. So thank you for listening, and I'm really happy that you were able to come, and I would welcome any question. Thank you.